with my project being in the chemical engineering department under Dr. Ryan Summers, with my work specifically being on the regulation of gene expression by the twister sister ribozyme. To begin, I'd like to help visualize and explain some of the terms that I'll be using throughout this presentation. First, a ribose switch. This is actually an mRNA fragment which regulates gene expression through conformational changes. This is actually the end goal of our research, and it's what the ribozyme is actually planned to be implemented into. Now, to help visualize what a ribozyme does and what a ribose switch really is, draw your attention to the two graphics, ribose switch off and ribose switch on. In the first graphic, labeled ribose switch off, you'll see that the blue RBS, the ribosome binding site, is actually blocked by our twister sister ribozyme. They're pictured with the shape labeled twister sister. The ribosome, the blue oval, is actually unable to interact with its corresponding RBS, and the green GFP gene is not expressed. The ribose switch is off. However, when the twister sister ribozyme is cleaved, a conformational change actually allows the RBS to be accessed by its corresponding ribosome. This allows the GFP gene to be expressed and the ribose switch is on. Now, the two most important points here is that self-cleavage actually produces two distinct bands. As you can see in the ribose switch on graphic, there are two distinct segments. Furthermore, self-cleavage is what actually frees the RBS and allows for gene expression. This is a very powerful tool for actually controlling all types of gene expression, as the GFP gene can actually be substituted for any variety of genes of interest. Now, let's take a look at the twister sister ribozyme specifically. This is actually a novel class of ribozyme, similar to the previously discovered twister ribozyme. However, it possesses an extra stem. Now, the important segments to take a look at here are the P1, P3, P4, and P5 segments pictured here. The other item that's of note is the cleavage site of the twister sister ribozyme, which is pictured here with the CLV. Now, the important thing to ask is the twister sister ribozyme self-cleaving in vivo, or in living cells, as this would allow it to actually be useful in gene expression, as mentioned earlier, in a ribose switch. Now, to begin, we actually used an in vitro cleavage test, self-cleavage test, excuse me. And this just simply means not in a living cell, and it was done as a precursor to moving to in vivo testing. Now, this was actually initially done through PCR amplification of our DNA template. We then used a T7 transcription to actually produce our twister sister ribozyme fragment, where, as you can see, the RBS is actually not free. There is the anti-RBS. However, with a cleavage assay where magnesium was actually added, magnesium is important in this case because it is actually a cofactor to induce the self-cleaving activity that we want the twister sister ribozyme to have. And after this cleavage assay, you can see that we expect to have two distinct bands. Now, to actually confirm whether or not this result actually happened, we would run this resulting sample on an RNA urea page gel, which you can see the results of here. As you can see, there we do actually have the presence of two distinct bands. To help visualize this, if you look at the P1, that red arrow there, you can very clearly see that we have two distinct bands, which is, as I mentioned, what we were looking for as our positive result. We were actually able to confirm the sizes of these bands by comparing them to the standards in purple, which actually are able to determine the base pair size of these nucleic acid samples. All experimental fragments of our twister sister ribozyme were cleaved. As you can see, the P3, P4, and P5 were cleaved as well. Now, what this shows us is that the twister sister ribozyme does express self-cleaving behavior during transcription. And these results actually led us to move to our in vivo self-cleavage test. Now, for this test, we actually introduce our ribozyme into the vector containing, as I mentioned previously, the GFP gene. This vector was then transformed into E. coli cells. These cells were cultivated and then were analyzed for fluorescence. This is important because self-cleavage in this case controls the expression of the GFP gene and actually will then show fluorescence. Essentially, we are using fluorescence as a measurement of self-cleaving activity. Again, as mentioned previously, magnesium was the cofactor used to actually assist with this cleavage. Here are the results of the in vivo testing. We can see from the P1 fluorescence graph that there is actually a distinct lack of fluorescence for both the P1 construct with and without magnesium as compared with the positive control. In this case, the positive control being the GFP gene being fully expressed. And what this shows us is that complexities in vivo are either inhibiting our ribozyme cleavage or our GFP expression in some way. What this led us to do is actually construct additional ribozyme constructs. The TS1, which is the construct that you saw on the previous page, was also tested. However, we created additional constructs, including the RMS TS1 through 4, as well as the TS4, which is different, and I will explain in a moment. The two pieces to consider when looking at these additional constructs are their GC content and their length. These are the two most important factors. To think of the GC content, it's best to just visualize that the higher the GC content, the more stable the ribozyme construct is. However, in our case, when looking at self-cleaving, we don't want these ribozyme constructs to be overstable, as this could actually interfere with our testing. The other aspect is the length of the ribozyme construct, and this simply has more to do with how the ribozyme will actually fold and interact after its cleavage. Now, as I mentioned, the TS4 construct, this is actually from a paper. This we pulled from a paper to actually use as a control. However, we did add, if you can see, the black loop there for in vivo testing, as in the paper it was used in vitro. Now, with these additional ribozyme constructs, they were tested, but the RMS TS1 was actually the most promising construct and thus the construct of interest for us. Now, to test this, we used a fluorescence time curve where we actually grew these ribozyme constructs in the E. coli cells in vivo and actually measured their fluorescence over time. This was also compared with their optical density, normalized with their optical density to account for cell concentration. Again, as I mentioned, fluorescence is being used here as a measurement of the self-cleaving activity. And we can actually see that fluorescence is seen for the TS4 and RMS TS1 constructs when compared with the PET28 empty vector. The PET28 empty vector simply serves as a negative control, as there's no fluorescence to be expected from this empty vector. Now, what these results show is potential self-cleaving activity for the RMS TS1 ribozyme construct. And this actually leads us into our future work. We are working to continue characterizing the RMS TS1 and the other ribozyme constructs, as I mentioned. We're also looking to use these ribozymes to actually produce a fully functional ribo switch, as I mentioned on the previous slide. Now, this image here is a great example of what that fully functional ribo switch could look like in a more practical usage. In this case, the GFP gene's expression is actually controlled by the ligand-binding aptamer, in this case with theophylline. Theophylline is actually used to turn the ribo switch on and off. Finally, I want to talk about ribozyme significance, as we've been talking a lot about metabolite-dependent gene regulation, as you can see there as the third biochemical process. But these ribozymes have a variety of uses in the biochemical world, including peptide bond synthesis, as pictured there on the right, as well as processing repetitive RNA sequences and many other types of RNA control. There's also a very large knowledge deficit in this area, as the discovery of new ribozyme classes has been extremely slow, and most ribozyme candidates' functions are currently unknown. So there's a high need to continue to do research in this area. I'd like to acknowledge my graduate student mentor, Xian Zhang, as well as the NSF for funding. Thank you, and are there any questions?
What do you think is the most important reason to continue working in the field of ribozyme discovery and characterization?
Yes, so as I mentioned, the ribozymes that we have discovered and characterized have very important biochemical functions, but there's so much we actually don't know about a lot of ribozyme candidates. So it's very suggested that these ribozymes that we haven't fully characterized could have very important biochemical processes that we simply don't know about yet. So there's a lot of potential.
Is there a way you control for the concentration of cells when measuring their fluorescence?
Yes, so this is what I was referring to when I was talking about the optical density. So what we would actually use is that AU there is actually a measure of fluorescence, and as you can see, that's actually divided by the optical density, which is used as our measurement of cell growth. So this fluorescence was actually taken into account with our concentration of cells. So this is all normalized data.
So you mentioned that the RMS TS1 construct was better than the other ones? Why in particular was this?
Yes, so what we're looking at right now, again, is that GC content and that length. That's what we believe to be the primary differentiating factors between these constructs. And if you look at that GC content for the RMS TS1, that is pretty low, especially when you compare its length is also relatively short. And we think that that low GC content actually allows it to be more flexible and useful in that self-cleaving context. We're still working to understand with some of these other constructs how they work, but it is believed that it's primarily the GC content and the length that is actually differentiating these constructs.
So, Ian, I think you've been working on this for over a year. Looking back at this project, is there one thing that kind of excites you the most, or what result or insight you gained that was a big takeaway for you?
Well, I mean, it's been super interesting just to learn more about, like, I guess you could say what's actually going on underneath the surface of these cells. I mean, like you said, I've been working on this project for a while. I mean, when I came in, I really didn't understand what was actually happening here. And now to be able to fully explain and articulate what we're actually looking at, as well as some of the potential applications of these riboswitches, I mean, that's been the most rewarding thing, is seeing where the progress is going and the impact that it can actually have.